Absolutely. Thank you, Amy. I'm going to share my screen. I hope you're all able to see my screen now. Thanks so much for joining us today. And good evening. My name is Elias Allara. I'm a clinical epidemiologist at the University of Cambridge and public health doctor. And today I'm going to talk about how polygenic risk score can be used in research and healthcare. And more specifically, I will be focusing on the case of iron, which is the focus of our research. So first of all, why is iron important? Firstly, because iron is crucial to life. For example, is necessary to, it's a very important component of hemoglobin, which is the protein that carries oxygen in the blood and brings oxygen to our tissues. And the imbalances of iron are extremely common, unfortunately. There are over 2 billion people in the world that are affected by iron deficiency. And there is some evidence suggesting the potential involvement of iron in several infectious diseases and diseases like the heart and vessel. We think that genetics can help reduce this burden of disease by providing insight into the health consequences of iron and by providing insight into the causes of iron disorder. Before we move on to the specific topic of our research, I'd like to introduce you to some terminology that will be useful to understand the next slide. First of all, by genetics, we mean the study of genetic information. It's also called the genome. A genome essentially is composed by long strings of code in the form of DNA or deoxyribonucleic acid. The genome is packed into chromosomes. We usually have 46 paired chromosomes, 23 from the mother and 23 from the father. The human genome in each chromosome is composed by 2 strands, and each strand consists of a sequence of 4 nucleotide bases that are complementary to each other. These are called adenine, thymine, guanine, and cytosine. You can see that they are paired to each other. So adenine is coupled with thymine and guanine is coupled with cytosine. And here we have the 2 strands that I mentioned before. A little bit more information about the term that we are going to use in the next slide. If we take the left strand from the image, which is this ugly red scribble here, the sequence will read from here. We would have a thymine, and then a cytosine, and then adenine, and then cytosine again. And it will read something like this. Usually, we take the first letter of each nucleotide for brevity. And we will ignore the other strand at this time because it's not it's complementary to the first one, so it's a little bit redundant. If we replace the first G in position 8, this one, the guanine, then we had a thymine in the pair chromosome. This is how the sequence will read. You can see that there is only one change in this position at this time. In this case, the underlying nucleotide in position 8 would exist in various forms, at least G and T in one individual. And we will call this mutation polymorphism, which comes from a Greek word that means things with various forms. Another term that we are going to be using in the slides is allele, which are the alternative version of the nucleotide. In this example, G and T will be allele for the polymorphism in position 8. And finally, in population genetics, the allele that serves as a reference when comparing different people, different groups, is called the effect allele. This could be either G or T, it doesn't really matter. It's usually just for a convention, the rarest allele. We don't have this information here because we only have information from one individual. But if we had information from 1,000 or 10,000 or 1 million individuals, we will know which of the 2 allele, G and T, is the least common. Another important characteristic of polymorphism is that contribute to the observable differences between humans. Let's see one example here. We have one observable characteristic, for example, blood cholesterol. And here in this plot, we have the distribution of this blood cholesterol in 3 groups of people. In the blue line, identifies the distribution of cholesterol in people that have thymine, TT, in both chromosomes. And you can see that this is the group that has the highest, it looks like already here, the highest average blood cholesterol, because the peak is the falls on the highest level of blood cholesterol. Whereas the people identified by the red line here have the lowest average levels of blood cholesterol, and this will be the people who have guanine in both chromosomes, and the group of people that has thymine in one chromosome and guanine in the other chromosome would have a distribution with an average that falls more or less in the middle between the two. And if we repeat this process for all the polymorphisms that we have in our study, can be 30 million or even more, then we could plot a graph like this. On the x-axis, we will have the chromosome, we have the position of each individual polymorphism. And on the y-axis, you will have the strength of association between each effector allele and the observable characteristic. So the higher the dot here, the stronger the association. And you can see that, for example, we have a lot of strong association here on chromosome probably 19, and also here on chromosome 5, on chromosome 2, and chromosome 1. And we did something similar in our work. We focused on common iron trait in over 240,000 participants. And we found association with 56 genetic areas, loci, of which 46 were not found before in previous genetic studies, which is kind of interesting because it can help us understand the causes of iron disorders. It can understand the genetic architecture of iron and so on. We also found that the overlap of loci across iron trait. And some of them, like this tree in the middle, were observed for all the 4 iron traits that we analyzed, suggesting that they may play a key role in iron regulation. However, we don't have that amount of information for all the traits related to iron. For example, we don't know much about hepcidin. Hepcidin is a key regulator of iron and is kind of at the center of iron regulation. It essentially suppresses iron absorption from the intestine and from red blood cell. However, we don't know much about the genetic polymorphisms that are associated with it. Was a study was published 7 years ago now that was actually quite a good study and analyzed more than 30 million polymorphisms, but they couldn't find polymorphisms explaining a large proportion of the observable variation in hepcidin. And the researcher who made this study mentioned that possibly this is because hepcidin may vary more in response to environmental factors rather than genetic. But we still don't know much. It's likely that there are some genetic polymorphisms associated with hepcidin. We still don't know which one. And let's move on to polygenic score finally. First of all, what are they? The word comes from the Greek word poly, which means many, and genes. Essentially, polygenic score indicate a score that represents individual genetic predisposition or risk of having a particular characteristic. For example, having a high polygenic score for diabetes means having a high genetic predisposition for diabetes. It's all about the possibility. It's not certainty. A person with a high polygenic score for diabetes may not end up developing diabetes. For example, if they do physical exercise or eat healthy diet and so on. So it's all about possibility. And how are they calculated? Just to get an idea of what they really mean. First of all, we select the polymorphism associated with the exposure of interest. For example, iron trade, in our case, is the same image that I showed a few slides ago. And we take the top association from our genomic study. Then for each individual in the data set, we multiply the product of the number of effector allele for each polymorphism with the effect size, and then sum this number. It's a little bit of a mouthful, but hopefully this example will clarify what I mean. This is one example using only 2 polymorphisms from this table and 2 participants. And participant A, the first polymorphism, we have 1 copy of the effector allele. You can see we have a 1 timing here, and the effector allele is timing. Where in the second participant here, in participant B, we have 2 copies of 1, which is not the effector allele. So we have 0 copies of the effector allele. For the second polymorphism, participant A, we have 2 copies of the effector allele, 2 cytosine. And for participant B, we have 1 copy of the effector allele. So we move on to this table on the right-hand side. And essentially, we multiply the number of copies of effector allele that are here by the effect size for each polymorphism. So in this case, for participant A, we multiply 1 by 0.031 and 2 by 0.037. And then we sum the 2, and we have the polygenic score. And if you compare the polygenic score between participant A and participant B, you can see that participant A has a polygenic score that more than twice as large as participant B, suggesting that participant A may have a higher genetic predisposition of for the trait of interest. It could be iron, could be something else. And we can apply this polygenic score, we can make them useful in biomedical research. There are at least a couple of ways by which they can be useful. Firstly is by helping us in risk prediction and stratification. The first step of this is to divide participants into equal parts based on the polygenic score. In this example, participants are classified in 5 categories according to a polygenic score for hypertension. And then we predict the risk for each part. You can see at the right-hand side of this image the risk of hypertension for people age 80 is very high for people in the highest category of polygenic score, people who have on average the highest level of polygenic score. And it's over 80%, you can see here. Whereas people that have the lowest polygenic score at age 80 have a risk of hypertension that is probably around 30%. So it's quite a big gap between the highest and lowest group of polygenic score. And obviously, if we can classify people this way, we can tailor intervention, can be behavioral or pharmaceutical, better to their needs. And also, we can use polygenic score to understand the causes of diseases. We can use them to estimate the association between the polygenic score and the risk of particular disease, for example, hypertension in this case. In this case, however, the polygenic score usually based on much fewer polymorphisms. that need to have a plausible role in the biology of the trait of interest, because we are interested in causality, not in prediction. There's an important difference. One way to do this is by conducting a Mendelian randomization investigation. Mendelian randomization is conceptually similar to randomized trial. It's different because Mendelian randomization is fundamentally observational in nature, so we don't really randomize people to have a higher or lower polygenic score. Whereas in a randomized trial, we actually allocate people to different groups, but it's similar in concept. So in a randomized trial, people are randomized into usually the treatment or control group, and the risk of the levels of the risk factor will vary in the control and treatment group. Usually it's higher in the control group because they are not getting the treatment, and lower in the treatment group. Similarly, the levels of the outcome of interest can be hypertension, can be heart disease, can be diabetes. It tend to be higher, ideally, in the control group compared to the treatment group. In Mendelian randomization, people are randomized, to say, by genetic variant or by polygenic score. If we focus on one single polymorphism, you could have, and assuming that the polymorphism is good, is protective, then if the polymorphism is present, we will have lower levels of the risk factor and lower level of the outcome. And the opposite would happen if the polymorphisms were absent. And we can perform a Mendelian randomization study using an approach that, at least at the beginning, is very similar to the approach that we use to build polygenic scores. Firstly, we select the polymorphism associated with exposure of interest, can be iron trait. But in this case, we are interested in those that are strongly associated and possibly likely causally associated with exposure of interest. And then again, we select the top association. And for each of these polymorphisms, we get their effect on the exposure and their effects on the outcome. The exposure, again, can be iron trait. The outcome can be any disease we are interested in studying, and hypertension, heart disease, or diabetes, for example. And finally, we estimate the genetic effect on the outcome per each 1 unit increase in the genetic effect of the exposure. As then just in concept, we plot something like this. On the x-axis, we have the genetic effect on the exposure. You can see for each dot represents one polymorphism from this table. And on the y-axis, we have the genetic effect on the outcome. And we can see that on others, the higher the genetic effect on the exposure, the higher the genetic effect on the outcome. And we are interested in estimating the slope of the red line here that tells us whether the genetically predicted effect on the exposure is associated with a higher or lower risk of the outcome. However, Mendelian randomization studies have an important potential issue, which is called pleiotropy. Pleiotropy is, in a stringent definition, means that one single polymorphism is associated with more than one trait. And as genomic studies become larger, this is becoming more and more frequent. And pleiotropy may be fine, may be okay, if the associated trait can be on the causal pathway to disease. It is called vertical pleiotropy because it goes vertically from the genetic polymorphism to the first trait, and maybe iron, and to the second trait, so cholesterol, and finally to the disease, like heart disease. However, pleiotropy can also be horizontal because the two traits associated with the polymorphism can be on different pathways. In this case, we have the same genetic polymorphism like before associated with iron and cholesterol, But in this case, the pathway that leads to heart disease does not go via iron. So in this case, if we look only at the association, we may incorrectly conclude that because the genetic polymorphism increases both iron and cholesterol and heart disease, therefore iron may cause heart disease. But this is not the case because as you can see, that this process goes through different pathways. So at the moment, in a very short summary, we have some studies showing that genetically predicted iron levels are associated with lower risk of coronary heart disease and hematologic traits such as anemia, and higher risk of stroke and infection. However, these studies have some limitations. For example, there are several biological pathways involved in iron regulation that could not be considered due to the lack of updated genetic studies. And the second limitation is that when using more polymorphisms, some association with disease may not be very reliable due to pleiotropy. So the truth in actuality is that we are not quite sure yet about this result. But we are working on it. We are working to improve our understanding of the genetic architecture of less common iron traits such as hepcidin, mentioned before, and investigate the role of iron-related biological pathway and systemic iron status on human health. So in summary, genomic studies are powerful tools to identify determinants of iron regulation and contribute to understanding the causes of iron disorder. Polygenic score and Mendelian randomization approaches can be useful to understand the consequences of iron on disease. And finally, there are ongoing efforts that are expected to improve our understanding of emerging iron traits, such as hepcidin, and the health consequences of iron. I'd like to thank all the researchers who contributed to this work. Colleagues from the University of Cambridge, as well as our funders, the NIHR and the British Heart Foundation, colleagues from Radboud UMC from the Netherlands, from Imperial College London, from the University of South Australia. And I'd also like to thank all the study leaders and participants that contributed to this study. Thank you. Very happy to take any questions you may have.